Hello students, welcome to George Biology. In the last session, we have gone through microtubules. Now, its structure is very important when you are going with centrosome as well as cilia and flagella. So if you haven't gone through that video, just check onto that video. I'll place that one in the description. Go with that one so that it helps you to get the composition of this centrosome topic. Let's come on to the centrosome. Now, if you look at the centrosome, it's a pair of centrioles which is connected by centroplasm. So there are two tube-like structures are present which are called as centrioles, and these two tube-like structures, they are attached perpendicular to each other for the formation of centrosome. So what is a centrosome? It's a pair of centrioles. It's a pair of centrioles which is connected with kinoplasm, or else it is also called as centrosphere, or else it is also called as centroplasm also. Now what is this centroplasm or else kinoplasm? It is a amorphous material. It's a powdery or else amorphous material which is present outside to the centriole, so we call it as pericentriolar materials, which helps in joining the two centrioles together in a perpendicular fashion. Now whatever this pair of centrioles are present, this pair of centrioles, these two centrioles together, they are called as diplosomes. Now where these centrioles are present, or as this centrosome is present, it's a characteristic feature of animal cell. It is also observed in algae. It is also observed in some members of fungi also. It means in algae, in fungi, as well as in animal cells, it is present. Then where it is absent? It is absent in case of plant cells. Do remember, based on the presence and absence of centrioles also, we can classify the cell division. During the cell division, if centrioles are present, that is called as centric mitosis. If they are absent, eccentric mitosis. It means, what is a good example for eccentric mitosis? Plant cells. In case of plants, that is acentric mitosis, where there is no formation of centrioles that takes place. Okay, let's look onto the structure of centriole. So we said centrosome is made of centriole. So if you study about centriole, that only will be the structure of centrosome also. But in twice you have to take that one in a perpendicular connection. Now this centriole, it's a non-membrane bound structure. It's a non-membrane bound. It's a non-membrane bound structure. How it is made of? It is made of 9 peripheral triplets. So let's look at the structure. This is one triplet made of 3 microtubules, 2, 3, 8, and here 9. So totally 9 triplets we have taken. So how many triplets are present on the peripheral side? It consists of 9 triplets. 9 triplets. And if you look at the center region, it consists of a protein hub. A protein junction is present, and that is considered as a proximal region of the centriole. So at the center, it consists of hub. So here 9 triplets are present, and at the center there is no microtubule which is present. That is why there is 0 microtubules at the center. 0 microtubules. Due to which this organization is called as 9+0 organization. So presence of 9+0 organization is a characteristic feature of centrioles. Now whatever these triplets are present, in these triplets, all the triplets are not complete ones. So let's say I will give some naming for this one. So at the inside one, the one which is near to the proximal side, I will take it as A. To the peripheral side, I will take it as B and C. So why we have taken like this? So this triplet, it consists of 3 subfibrils, which are termed as A, B, C, where A, that indicates the first subfibril, which is a complete ones. Why it is considered as complete? Why? Because it consists of all the 13 protofilaments, which makes a perfect microtubule. Now this B and C, now these subfibrils, they don't have a complete composition. It means one or two less protofilaments are present in making the complete microtubule. It means it is a incomplete ones. Incomplete microtubulitis. Now these triplets whichever are present, they are connected to the hub by presence of radial spokes, which are made of protein radiating from the center and connecting the triplets to the peripheral side. It means how many radial spokes are present? 9 radial spokes are present. So let's detail everything here. It consists of 9 triplets which are present towards the peripheral side. Number 2, it consists of 9 radial spokes. Radial spokes are present. Number 3, at the center, it consists of hub. How many are present? 1 centriole, 1 hub is present. A protein hub is present. Or else it is also called as central hub. Central hub. Now these alignment that helps in formation of a complete wheel-like appearance, which is called as cartwheel appearance. Cartwheel. The cartwheel appearance of centriole is due to 9 radial spokes, 9 triplets, and 1 central hub. Altogether, they help in formation of a cartwheel appearance of centriole. Along with all these things, these 9 triplets, they are freely present as of now. But to make a complete tube of centriole, they have to be united. So that is why the A subunit and the C subunit, they are connected by a linker, and this linker or a bridge, it is called as AC linker. A subunit and C subunit of all the triplets, they are connecting here. So we call it as AC or else CA linker. What is its function? Because of this one only, all the triplets are united to form a centriole. It means it is helping in making the structural geometry of the centriole. So what is its function? It helps in making the structural geometry of the centriole. Apart from this one, to the peripheral side of each triplet, it consists of certain amorphous material is present, and this amorphous material which is present towards the peripheral side of all the centrioles of all the triplets, it is called as maculae, or else it is also called as pericentriolar material. So how many maculae are present for every centriole? That is 9, which are present here. So here it is called as maculae, or else it is also called as pericentriolar material. Pericentriolar material, or else it is also called as microtubular satellite. Now what is its function? It helps in formation of microtubules. So it acts as a MTG, microtubular generator. That is its function. Let's go with the overall functions of centriole function. Now these centrioles, they help in generating the microtubules. It means they help in formation of spindle apparatus during the time of cell cycle. It helps in formation of spindle apparatus. Spindle apparatus. Number 2, it helps in formation of basal body of cilia and flagella in the eukaryotic cells. So here it helps in formation of a basal body. Basal body of cilia and flagella. So let's look at the questions. What can be asked here? The origin of cilia and flagella in eukaryotes is from. What is the answer here? That is from the centrioles, the origin that takes place. Number 2. What is the organization that you can see in the basal body of cilia and flagella? I'll give you options. 9+2 organization. Number 2, 9+0 organization. What is the answer? 9+0. Why? Because basal body of cilia and flagella in eukaryote is because of centrioles. how many microtubules that are involved in one centriole formation? How many you can see? At the center, there are no microtubules, so we have kept it as 0. On the peripheral side, 9 triplets are there, we said. It means altogether how many? 9 into 3 microtubules. That means 27 microtubules that are present. Remember, based on this, numericals also can be given. Let's take a question on this one. For example, in a cell, if it consists of one centrosome, how many microtubules that are involved in formation of that one centrosome? If a question is given, what will be the answer here? One centriole that has 9 triplets. It means every triplet has 3 microtubules. So one centriole, 9 triplets, each has 3 microtubules. So altogether it is 27. 9 into 3, 27 in one centriole. But a centrosome consists of 2 centrioles. So 27 plus 27, that is 54 microtubules that are present. That is how you can frame the question. So this is all about the centrosome. In the next session, we'll go with cilia and flagella.